Okay, hello everyone again. Welcome to another Saturday live stream. I'm again, like every time, doing a little bit of housekeeping here to see if everything works. It seems to be working. Yes, here we are again. And yeah, you can already see I put a little worm here under the microscope. Want to explain to you a little bit of how it actually, why it's there, from where I have it. Yes, a little bit of an organizational thing for today. Today's live stream, I have to make it a little bit shorter, maybe maximum 1 hour, not 1.5 hours, because I have to go somewhere. Right, so today's live stream might be a little bit shorter today. It doesn't matter. I still hope that I'm able to show you some cool things. Yeah, and I would like to use the first couple of minutes very quickly to say hello all around the world. I'm now not going to read out all of the people who've joined in now yeah, in the chat, just to keep it a little bit shorter. And I just want to tell you a little bit about this here. This Iolosoma, that's the name of the worm. It's an it's a segmented worm, and I got it from a jar, and maybe you a few weeks ago you've seen the video where I put a Daphnia, which is a water flea, under the microscope. Let me take my glasses off here. This Daphnia, the water flea, was grown over with those tiny rotifers. Yeah, those wheeled animals, animalcules. And yeah, they are not doing well. As expected, the population dropped, so the rotifers yeah, seem to have yeah, taken down the population of Daphnia, but then those worms started to multiply quite a lot. So I've got now a whole jar of, I don't know, with hundreds of these Iolosoma worms. There are actually more of them, but I don't know where they've gone. This is basically where I said, wow, I'm just going to show this to you as well. I actually had several of those here on this microscope slide, and I don't, here maybe they are. Here more, right? Yeah, and they're basically have been reproducing quite a bit. And yeah, and what I want to show you here is already a very interesting thing. So, I mean, these are some specimens that I always keep on coming back to. I've just realized that the exposure and the frame rate of the camera is not very high. But if you actually look over here, where's this? Maybe the other one. Yeah, I could see it a little bit here. Yeah, here it is, this one. I'm going to switch on the arrow to show you. Do you actually see that there is over here a new head developing right in the middle of the worm? Because these worms like to fragment. So what's going to happen is that yeah, the head will continue to grow, and then this is going to be the new head, and then the worm's going to split, and then you've got two worms. It's a form of asexual reproduction, right? That's something that they like to do, right? So you can actually see over here this a head growing right in the middle of the worm here. Yeah, and this takes maybe a couple of days, but I've been observing this for some time, and this is kind of cool, right? So yeah, if you have just joined in this type of live stream, if you have any questions or so, please feel free to ask them. But again, today, if you just joined, I have to keep the live stream a little bit shorter than in the previous than my previous ones, because of yeah, I've got to go somewhere, okay? So this is actually, we're just going to look at them a little bit longer. And then a few other things I would like to show you today. It's a little bit of a, how do you say, a special request. Just the new school year started. And yeah, also the first biology lessons. I'm a biology teacher. I've had my first biology lessons, and then of course word was spread quite quickly that also I'm making microscopy YouTube videos. And then I asked my students, what do you want me to put under the microscope? Yeah, and then one of the students says, well, can you put some a watermelon under the microscope? So I've actually got a piece of watermelon here, right? Which I would like to put also under the microscope. And yes, I also have some cheese that I also would like to show you, because we can find bacteria and fungi on them. So it's a safe source of microorganisms, okay? Yeah, so I see that more and more people are joining, yeah? Okay, Eversmall is asking, did you get the reflected light images I sent via Dropbox link of the sensors? Oh, thank you for asking. I think I did not. I'd have to check this. Maybe I did not see the email. Okay, ever smaller Jesse is yeah. But thank you for reminding me. I'll have a look. Okay, because yeah, I'll have a look at this. Yeah. So, okay, well, another thing I just want to show you is the following, is that it's possible actually to look into the digestive system. So what is also possible to do, and I've done this already also before, is to feed those worms, and then you can actually see how those worms are taking up food. Yeah. Let's go up with a magnification, because Iolosoma, the name of the worm, is basically also known as the so-called the oil droplet worm. The reason is because, maybe I have to open the condenser a little bit here. Okay, you can actually see that the head of the worm has those red dots. Yeah, and yeah, these are apparently oil droplets, and yeah, they're pigmented. Yeah, so if you just look here, there are these dots on the surface of the worm, and yeah, apparently these are oil globules, fat droplets, fat globules. That basically yeah, are characteristic for this worm. Yeah. So there are yeah, quite a few of those. These are kind of very common ones, and I think they're kind of cute. As a matter of fact, it was actually this worm here, this very worm that I made a couple of years ago, a YouTube short video that really took off and went viral. I got a lot of subscribers by just showing this worm, and then basically my views crashed down again. Yeah. What are the clear blister looking things? Okay, yes, I understand what you mean, right? And I've noticed them as well. I'm just going to show you. Sometimes over here on the side of the worm, there is this blister that you see, and I think I know what it is, because, and yeah, here, do you see those? Yeah, those blisters here. And I have a hypothesis, and I don't know if this is correct, but that's something that can be tested quite easily. I think this is because when just a few minutes ago, when I put the cover glass on top of the worm, I might have had a little bit too little water, and I might have squeezed them a little bit too much. So I suppose that's some kind of it's a hypothesis that I have. I suppose it's an injury that the worms have because I compressed them a little bit too much, and then I added a little bit more water to give them a little bit more freedom of movement. But this is actually something that I assume. Okay, it might be an injury, but in order to find that out, I'd have to maybe put some other worms under the microscope. Yeah, here you see one. Yeah, quite nicely. Yeah, yeah. But if there's actually a head growing out. Yeah, yeah, like a hernia. Yes, yeah. That, again, I'm not sure, but I also did notice them just a few minutes ago after I basically put them right under the microscope slide. I said, "What are these blister-like things here?" And I think this might be an injury. Yeah, yeah. And here again, you can see very nicely over here a new head growing right in the middle of the worm. Yeah. One thing that I've also, and that's a little bit difficult now because they still have a lot of movement, but let's go up with the magnification yet further. It is actually possible, if you're lucky, to see the muscles that are attached to the hair on the side. Yeah, because they are actually using those hairs to push themselves off. And I once, some months ago, some years ago, I made a really nice video basically where you could actually see those bristles moving forward and backward by the muscles that pull them. Yeah, yeah. So occasionally I will now answer again. If you just joined in today's live stream, I have to make it a little bit shorter because I have to go somewhere, but I wanted to keep the weekly schedule a little bit. So if you have, there's a Maradona is asking if I have a vase, the tap water has been waiting for 3 days and the water is cloudy, what can I find in there? Cloudy water always indicates, or most cases indicates, some kind of suspended particles, and if it's not sand, or then most likely it is indeed microorganisms, can be bacteria and or other ciliates and whatnot. Yeah, cloudy water is always an indicator that you can actually find something in there. Usually, it's also like this that microorganisms, because in many cases they are more dense than the surrounding water, they will also tend to sink down a little bit. Yeah. How did you manage to place that cover slip on without turning the specimen into microscopic pancake mush? Honestly, this is almost what happened. You have to add sufficient water. When you add a lot of water, maybe sometimes a little bit too much water, then the cover glass actually is going to float. And yeah, I can actually show you how this actually works. I'm going to go down with the magnification a little bit more. And if I'm going to add now yet more water, and this should actually, yeah, show you that the worms are actually able to move yet more freely. Okay, let's see. The water is soaked in, and this actually will make the worms now a little bit less wide because they're not pressed flat anymore, right? But yeah, and then they should also be able to move, yeah, a little bit faster. And occasionally also something nice. Look, this worm here was pooping a little bit. You can actually follow the movement of food through the worm. What I've done some time ago is I've actually been feeding it some starch grains from a potato. Those starch grains are very polarizing, so this means optically active, and then you can actually see them actually quite bright and very well inside the digestive system, how it actually moves through the digestive systems, and then the worm poops it out again undigested because they're simply too hard. Yeah. How can I find amoeba? Well, first of all, amoeba, you've got to be lucky that there are enough in there. Chances are pretty good that there are amoeba in there, and you've got to be patient because amoeba, even if they move around, they don't always move very fast. So what I have a suggestion is if you have a water sample, just make sure that you don't take the clear water, but that you take the sediment, so some of the a little bit solid material as well, and then make a video. And then you speed up the video, and then you're also able to see all of the movement. Amoeba are present, might be present, but they might be difficult to see because they don't move very quickly, and therefore they can be overlooked quite easily. Yeah, yeah. So this is again very nicely. You can see now the head much better, right? So this is again, this is going to be the worm is going to separate over here, and then you're going to have two worms. Yeah. So. Yeah, exactly. Dead leaves, sediment, some solid material. What I've also done is you take some solid, I don't know, dead decomposing leaves and carefully scratch the surface and put some of that stuff under the microscope. Of course, this is also going to damage some of the things on there, but that's okay. Yeah. So yeah, so this is kind of fascinating. I was quite a little. Yeah, I'm just going to show you the jar again. Maybe some of you who've joined in a couple of weeks ago, you remember this. That's the wrong microscope. You remember this jar here with some Daphnia, and remember that some of the Daphnia. There might still be a few in here. And there are a few here, and those Daphnia had those how do you call them, those rotifers on them. I don't know if I'm able to now, because there are not so many of them here. I don't know if I'm able to catch a Daphnia as well. Let's do this very quickly, and I gotta put on my glasses again, and let's see, and then we can just as a little reference. Yep, I got one. And now without a cover glass, let's see. So that's let's put it without a cover glass. So I'm just gonna, and where is the scope view again? So here we are with the worms, and let's have a look. Yeah, let's lower the magnification a little bit. Yep. Yeah. So if you kind of remember the video, the live stream from some, I think 1 or 2 weeks ago. Yeah, so you can actually see on this water flea that there are these tiny rotifers growing on it, and I think they have been kind of stressing the Daphnia population quite a bit. So the Daphnia really did drop down, even though I've been feeding them quite well. But if you add food, then of course you're also feeding the other organisms that are present. And right now, I don't know, hundreds of these tiny worms in there right now. Yeah, the Daphnia population now collapsed. Yeah. So you see that it's kind of like a natural progression of that's a water drop. Yeah, it's like a natural progression of organisms that we're able to see here. Yeah. So back to the worms again. Yeah. So here's one worm which is here are a few more. So those worms they keep on reproducing, and occasionally add some yeast, maybe some algae, and I also suppose because they're decomposers that they're also feeding on, of course, the dead Daphnia. Yeah. So I'm just going to read a couple of the questions here that you might have. Are the worms there visible to the naked eye? Yes, they are. They are the Iolosoma. You know what? I'm going to type it into the chat. A-E-O-U-O-S-O-M-A. So worms. So I'm typing this now into the chat in case you kind of also known as so-called oil droplet worms. And yeah, they are very very common and frequent, and kind of interesting to observe as well. I like them a lot because they are kind of transparent, and then you're able to see a little bit what happens inside the digestive system. But you know what? I just realized I'm going to not do the following. I'm going to put in now the phase contrast annulus does work like a, how do you say? Here we go. That's now dark field. So I'm now using the phase contrast annulus, as a dark field filter. It also looks kind of nice. So this is quite fascinating. I want to talk a little bit. I decided, of course, so just stuff under the microscope, like always. I just want to, because school is starting right now. Let me remove the arrow, and because I'm teaching, of course, biology, high school biology right now. I have a syllabus to stick to. But these worms are not part of the biology syllabus, and I'm going to be now basically just sharing with you some thoughts that I have about my own biology lessons, or generally about the biology syllabus. And that is, I'm a little bit, how do you say? I'm not completely uncritical, because you see, we biology teachers have to cover all sorts of more or less advanced topics, cell biology and cell division and photosynthesis, cell respiration, also the biochemical processes that happen during cell respiration. Very abstract stuff sometimes, right? And biologically it's considered important. But the problem that I'm having is by teaching these things, I became a biologist because I was fascinated observing nature and the environment. And I just want to question a little bit the way that we're teaching science, because this fascination for nature, the love for nature, the wonder, the sense of wonder, we teachers have to be careful that by teaching the subjects that we love, we're not kind of driving out a little bit the sense of wonder, that at least I always have when I observe nature. Look at this. You can now see that the inside of the worm looks a little bit green. Okay, why is that? Well, because I've been feeding the jar, adding chlorella algae to the jar, and that's evidently the thing that they've been eating. Okay. Occasionally I'm just reading. Have you tried to do DNA-based identification of microorganisms for fun? This is a very interesting question. I can tell you the following: no, because I do not have the equipment to do that. I have been doing DNA-based identification during my diploma thesis. And it worked like this: that I was growing bacteria, and I was extracting the DNA from the bacteria, and I was basically doing a so-called PCR reaction and sending that then to the lab. And they were doing the sequencing for us because it's pretty specialized. They had those machines that were doing this automated. I got an email back with a DNA sequence, and which I could then type in, copy paste into the database. So basically, in order to do that, you need basically a sequencing machine. You need all of the relevant fine chemicals and so on. That is a cost issue, right? It's of course also a time issue. So for this reason, if you want to get stuff done like this, you basically normally do the following: you contact a lab, and they will do that for you. You kind of outsource this as a service. So even inside our research institute, we were outsourcing these tasks to a department inside the university. Yeah, because it's too specialized and yeah. So it's an interesting point, really, because I think doing DNA analysis at home, that would be really cool. Yeah, because it's extremely powerful. Yeah? But not for hobby purposes, but maybe also not quite as fun, really, as doing direct live observations. Yeah, yeah. So I'm going to quickly, yeah. So yeah, ever smaller though. To be fair, my teacher was probably an escaped lab rat. She was horrible. I'm sure I would have at least liked it somewhat if we had nice and cool teacher like you. Well, thank you very much. Thank you very much. But I tell you as well a little bit. It's teaching biology or any natural science is not always easy for the reason that the stuff that we have to teach sometimes I envy a little bit the physics teachers and the chemistry teachers because it's much easier to do, how do you say, fascinating lab demonstrations. I mean, in physics, you can quickly, I don't know, make a few things, I don't know, and demonstrate electricity and get some light bulbs or LEDs lighted up and so on. In chemistry, you can do your, I don't know, your funny little explosions and chemical reactions. But in biology, it's a little bit more difficult. And microscopy is, of course, the one of the central things that you can do in school. But the problem is that, you see, the syllabus does not always allow that. I mean, when I talk about, I don't know, advanced stuff like glycolysis, which is a part of the metabolism, right? Glycolysis and the Krebs cycle and electron transport, all of this fancy metabolism. I mean, this is very abstract. There is nothing that I can really show. Yeah? For this reason, I sometimes as a biology teacher, I envy the other science teachers because at least they have an easier possibility, easier opportunities to do impressive demonstrations. Yeah, I like, for example, I don't know if you know the YouTube channel Action Lab. I like to watch that, yeah, because they're simple, not simple. These are sometimes pretty advanced, but fairly straightforward demonstrations that have a really nice effect. And in biology, we're a little bit limited with that. Yeah. So yeah, can you test these worms with a drop of vodka? I don't have vodka, but I have concentrated alcohol here. But you see, the problem is the following. I mean, I could add some alcohol here. It's going to kill them. I don't know if I should do that. Yeah. So that's something that I could do that. And honestly, it's not that I feel sorry for those worms, right? They have a simple nervous system. Maybe they're not even able to feel pain. But I'm not doing that out of principle. I have no problem adding alcohol to microorganisms like single cell like bacteria and paramecia and other ciliates. But I generally don't do that. I don't like doing that so much to multicellular animals, not because I feel sorry for them, but because how do you say this? I want, as an educator, I want to set a good example to the students who might be watching these videos. You know what I mean? Yeah. So what I recommend, therefore, is if you want to do that yourself, please do that and add a little bit of alcohol and other substances. Yeah, you see, yeah. How shall I say this? Yeah. I'd have to answer to people, why am I killing these cute worms? You know, it's emotionally not always very, how do you say, yeah, nice for some people to see. Yeah, yeah. Steve Mold, yes, I've seen. Yes, yeah. Honestly, I know Steve, not personally, but Steve Mold is. I always watch his videos. Yeah, so that is the yeah. I mean, you can add not only alcohol, you can also add hydrogen peroxide and so on. And yeah, on the other hand, I have to say I am a little bit against being overly ethical because some people are kind of worried too much about adding alcohol and disinfectants to microorganisms because they think killing them is not okay. I don't have problems with that. Yeah, yeah. So much. So you know what? I've been talking now for 24 minutes already. I want to move on. I think enough of those worms now, because again I want to how do you say this? Fulfill the wish of one of my students who asked me, "Can you put a watermelon under the microscope?" So I'll put a watermelon under the microscope. And you know what I'm going to do with those worms here? I'll just show you. No, I'm going to not kill them. So here, let's have a look again at the, how do you say, this here is the water flea, the Daphnia. So I just basically rinse them in here, knowing that they're gonna die anyway. But at least we don't have to watch that. Okay, and over here, those worms here. I mean, those worms don't really have an identity either, because I mean, they reproduce asexually as well, right? So let's open this here, yeah, and let's see. There is no worm on here, or is there? Okay, let's just rinse it off. Yep, gone it is. And over here, I just basically rinse off everything back into, so that nobody can complain about animal cruelty. No, honestly, people have actually already complained about that because I once mistreated a little single-celled paramecium, and I got some not so nice comments in my YouTube channel, and then I said, okay, if this stresses the people too much, I'm not gonna do that anymore. Yeah. So let's do the following. Yeah, so one of my students, as I said, is I asked, oh, what do you want me to put under the microscope? And then one of the girls said, a watermelon. And I says, okay, I don't know if a watermelon is the most exciting one here, but today I bought a watermelon, and it's not the first time I'm putting a watermelon under the microscope, because some years ago I did already, and I was able to see a couple of interesting cells and also some xylem tissue. I don't know if you're able to, yeah. And the nice thing about the watermelon is that it's a fairly easy specimen because it is so extremely soft. Okay, so and now I'm going to show you how to make a compression specimen. This is going to be pretty, how do you say, extreme. So yeah, so I have to make this small, and this only works with how do you say, with specimens that are fairly soft. Now I got to be a little careful, because the risk of this actually cracking is very high. So I have to make it a little bit smaller. Okay. So I think this is gonna be fine. Yeah. So this is the watermelon now, okay, a tiny amount. And now I have to carefully compress it here. Yeah. Compress it between the cover glass and microscope slide. You see, you cannot see it anymore, and any excess liquid I'm just gonna soak up. And that's it. Okay. Yeah, so you see it over here. We're gonna put this under the microscope, and of course, and we're gonna see if melons are also made of cells or not. Of course they are. So where's the scope view? Here we go. Ah, yeah, it's still a dark field. So let me put those worms away. Here we go. Oh, it's way too bright. Yeah, well, I don't know. Okay. Okay, looks a little bit messy, but maybe we're lucky to see, yeah, maybe there's some vascular tissue. Let's go up a little bit with the magnification here. Yep, the individual cells seem to be, yeah, like a dense network. And what I think, I was actually once lucky, maybe this here, I don't know, let's see, let's go up. Yes, I'm happy. I'm happy. This is actually educationally valuable. So this is actually educationally. What age range do I teach? I used to teach 10 to 18. I'm now teaching 17, 18-year-olds. So basically high school, the last 2 years of high school before they graduate. I teach an elective biology course, advanced biology course. This is what I want to show you, and if, I don't know, some of my students said that they might be actually also joining in into the live stream. So if these are some of my students who have also joined in, you have to know this actually. So what we see here is, do you see this spiral thingy here? Yeah, this looks like a spiral, yeah? That is called xylem. Xylem, xylem, and these are basically tubes that transport water, yeah, inside a plant. Yeah, can we see the phloem as well? Probably, but I don't know where it is. Okay, Yeah, so that's the thing. The phloem, it's yeah, I'd probably have to prepare to see it, but the xylem is really nicely visible here. And yeah, this basically are strengthening rings. So basically, there's long cells, but because there's water flowing in there, you have to. It should not collapse, right? Otherwise, if it's yeah, if you've got flowing water, then there's the danger that it actually collapses because of the pressure. Because if there's movement, moving water, then the pressure is actually lower, and so you have to make sure that there is basically the cell wall is strengthened. It's a little bit like this. We've got cartilage here as well, right? Those cartilage rings here on our throat. So when we inhale and exhale, that it remains open, yeah, because we've got moving air, and then there's also the danger that it kind of collapses. And the plant cells that transport water have the same thing here, right? So if you happen to see these spiral-shaped structures, sometimes you even see them, for example, in water. Why in water? Well, if there are decomposing plants in there, then yeah, those xylem is actually quite strong, yeah, and does not break down as easily. Yeah, and so for this reason, yeah, you can find those spiral-shaped structures quite frequently in nature. Huh? Yeah. Can I find stentor in the vase water? I don't know. You gotta try it. Probably not. Okay, and I will say you why probably not. I'm not saying not, I'm saying probably not, because you have to see if you have somewhere an isolated a jar of water or standing water from a flower pot or whatever. You can only have those microorganisms in there that basically have reproduced, and they have to come from somewhere. So unless you maybe have added some pond water with stentor already in it, those stentors will not automatically appear on their own. Okay, so that's what I kind of want to make clear. So generally in those artificial areas like flower water, standing water at home and so on, generally the biodiversity is not so high. The reason is because some bacteria and fungi maybe, or algae that happen to be around, they will start to grow there, but those larger interesting organisms often appear in nature, and they won't just appear there on their own. So if you want to do that, you actually have to get some pond water and enrich them yourself. Yeah, so you need to have those organisms there in the first place before you can actually grow them. Yeah. So that's simply something that I just wanted to clarify here. So don't be disappointed if you do not find a lot of interesting things in vase water, right? But do have a look. It's probably not very likely. Yeah. I find most stentors by scraping the film off of submerged plant stems. Yes, honestly, I've also gone to a pond once, collected a couple of water plants there, and it was full with interesting stuff, right? So because stentor being a so-called sessile, it likes to be connected, you need to actually have a surface to get it from. So you're probably not going to find it in free water. Yeah. Yeah, so this is, So let's go down a little bit again with the magnification, maybe to get a better overview. Yeah, yeah. So that is basically something that I was hoping. Now, you're probably going to say, where were the cells? Okay, that's actually a good point. I think I suppose all of those lines that you see here must be cell wall. I mean, a watermelon contains a lot of water. Yeah, so the cells themselves are difficult to see, I would say. Some red pigment that makes basically the watermelon red. Yeah, but it was actually how do you call this, the xylem that I really liked. There was actually something that looked a little bit like, sometimes maybe some of these round structures that you see here. Yeah, lots of look, it's a complete mess. Yeah. So what I assume, let me show have the arrow over here. Yeah, I assume that those round things that you see here, that these are individual cells. Yeah, and those lines that you see here are most likely cellulose from the cell wall. Yeah. So that's a little bit the thing for concerning the watermelon. Here again, xylem. Do you see those spiral? This is a long one actually. Let's go up with the magnification again. Yeah. So here again, you see this long xylem-like structure. I also found them in pumpkin. You find them in all over the place. Yeah. And looks like the cells are connected in a dense network. Do you know what this network might be? This network is most likely a cell wall, cellulose. Yeah, because the cells are connected to each other. Yeah, over the one cell connected to the next cell, and they're sharing a cell wall. Yeah, so this is most likely yeah, cell wall. That's a nice looking xylem. Look, that's a long tube. Oh, here's another one. Yeah, look, there are many more. Many more. Yeah. And different plants, actually, the xylem, sometimes there are separate rings that you have, right? And sometimes it's actually spiral shaped. So there are different types. Yeah. So was this the sample, the flesh? Yes, the red part. What about the green cells from the oh gee, you're asking something. But that's actually a good point, and I don't know if I'm able to do that. Let me quickly try that. It's actually a good point. You see those cells, red cells here, are quite easy because they're so soft. The question is, how am I going to be able to get a very thin layer from here? That's a little bit the question, right? I'd have to cut it. And I'm going to try the following way. And this might not work. I've not tried this yet. But maybe by kind of ripping it. No. You can do that with onions. If you do that with onions, then you can actually, yeah, this part over here on the side is actually sometimes very thin. With onions, this works much better. Kind of ripping it. But you know what? Why not try it in any case? But this is not going to be a lot. It's just this very fine edge here on the side. Do I have my fine scissor over here somewhere? You see, that's it's always a little bit difficult to make sure that it's thin enough. Yeah, so maybe we're lucky with this part here. Yeah. So with onions, you can actually do that. With red onions, you cut in and then you kind of put your thumb over it and you kind of peel off the layer. But here, that's not so easily possible. Yeah, because it's very brittle. But maybe we're going to be lucky with this one here. Just realized I need a little bit of water maybe to make a proper wet mount. So I do have water here. No, great. I'm super well prepared. I don't have oh, here it is. Okay. I need some kind of so let's add a little bit of water. Where is my pipette? Let me quickly. So, and of course, it's too thick. Let's give it a try in any case. You see, trial and error, that's what I just want to recommend. And it being green, of course, we hope to see maybe some green chloroplast. I am not very confident. Okay, but let's give it a try in any case here. Okay, the scope view, yeah. And then I want to move on to the cheese. Let's go down a little bit with the magnification here. Here we go. It's too bright. Where is it? Here we go. Oops. This is completely out of focus and it's too thick. Well, maybe, I don't know. We have to really find here on the side, couple of cells that, maybe over here. Here, yeah, that's better. Okay, yeah, here we can actually see the cells better. I need to explain this a little bit. They're not green, and I can tell you why. Because they're broken. They're broken open. Yeah, but over here, obviously, we've got here where it's so thin. Yeah, we've got, of course, individual cells. You see a huge, thick cell wall, right? Normally, you have the chloroplast green inside. I think the reason why they're not green is because as I ripped it open, the cells kind of broke open, and then the green chloroplast also ran out. It's much more green over here. Yeah. but it kind of shows a little bit. I mean, you can try this also with a tomato skin. And what you do is you kind of carefully scratch off all of the pulp of the tomato skin, and you're also going to see very nice cells. Yeah, but I just wanted to, I think the demonstration here is the following, that if you want to get very thin cross sections of plant material, especially if you've got some kind of a layer, you got to kind of rip it off somehow carefully, because otherwise because in many cases you're not able to cut so thin. Yeah. And so, yeah. I don't think that I can go up with the magnification here, because I might be touching the cover glass. Yep, I'm touching the cover glass. It's yeah, so that's not gonna work. Yeah, but actually I'm still quite happy. Yeah, so that's basically the surface of a melon. Yeah, well, it's interesting. Yeah, yeah. For me it's actually the first time that I'm seeing. Yeah, so thank you for the suggestion. Yeah, I've seen the pulp of the melon, the red part. I've seen, of course, many times before already, but yeah. This was also a first for me. The side issue again, a little bit about the history when they, back in the 19th century, 18th, 19th century, when they, basically when microscopes first were available and folks were looking at cells, they essentially, it's quite interesting if you go a little bit into the history of science, their microscopes were not quite as good. So what they could actually see were only the cell walls. So at the beginning, the scientists were really fascinated by the cell walls, and they could not really see the contents of the cell. Yeah, and there was at the beginning there was a debate, and they were actually thinking a little bit like that living things. Maybe the cell wall is alive. It's like a network of life, so to say, with holes between it. So like a sponge, right? But nowadays we actually know the cell wall itself is not alive. Really, it's the contents of the cell that's actually alive. Yeah. So yeah. So that's kind of the thing. Yeah. Now I need a paraffin mounting it. Yeah. Yeah. You need to paraffin mount a watermelon seed and microtome it. That's another thing. Gee. Yeah. You're really trying to set me up here for something, because I've been thinking about actually going a little bit into paraffin mounting and microtoming, but honestly, a really decent microtome actually takes up a lot of space, and I don't have that space. Yeah, and yeah. In any case, that's really something. Yeah, a proper microtome would be really nice. What you have to do then is you have to dry the specimen, dehydrate it, embed it in paraffin. And then and slice it very thin. Yeah? So I've used those hand microtomes. Yeah, oh, definitely, of course, I have them. But those hand microtomes don't give you very fine cuts, because I mean, not quite fine enough for. But if you actually really want to make paraffin mounts, I think some kind of rotation microtome or so would be better. Yeah. PG, no, I did not try PG, yet. Okay. And yeah. So what I'm going to do now is the following, is because as I mentioned, I do have to stop a little bit earlier today. What I'm going, and how many minutes now? Okay, 43 minutes now. So what I'm going to do now is I want to also put under the microscope some cheese, because this is also something that I wanted to show you. Yeah, I'm going to use this cheese over here, because this one is actually quite a nice one. Oh, I need to, how do you say, desk view. Yeah, so this here is a very, very smelly cheese. It's pretty good. I mean, I like it. Not everyone might like it. And this is a nice example. Yeah, this is a nice example of of bacteria and fungi growing on here. So as always, I need to add a little bit of water to the microscope slide here. Okay, and I'm now going to scratch the surface here. And because people sometimes ask me, well, I want to see bacteria, I want to see fungi, where can I get them, and can I grow them myself? And I say, no, you should not. because strictly speaking, if you are growing unknown bacteria, it's already a biosafety level 2. So if you still want to look at microbes that are safe, you have to use those that are used on food, right? And here, that's actually a nice example here. So what you do is you carefully scratch the surface a little bit, right? And until you have, and you suspend it in here. So of course it's not only bacteria and fungi here, but also some maybe some milk protein. So what I'm going to do is I'm just going to, all of those thicker parts, I'm just going to put it to the side here. Okay, I'm going to put this to the side here, and a cover glass goes on top. And then we go again under the microscope. Okay, so usually I want to probably wipe away this here. Okay, and let's have a look. Okay, because I highly recommend always to use only microorganisms from food, and not basically, you should not be spoiling things or food or growing them, because you never know what you're growing, and then that's not good. Yeah? So this is, I have to kind of keep on readjusting the brightness a little bit. So here we go. Okay, some air bubbles, so that's not the right place here. Okay, but yeah. I mean, this is honestly, I've already had a more dense network before, but it's good enough for our purposes. It's not quite as dense as one of my previous ones here, but let's go up. But I'm getting the point across here a little bit. Yeah, so, and then I'll explain what we're able to see here. Maybe sometimes it's even better if it's not too dense, because then we're able to see the things better. Yeah. So what we have here is the following. So that's those lines that you see here, these are fungi. You can also see the individual cells, and they are eukaryotes. Because you see that inside the cell there are, those cell organelles, could be a nucleus, a nuclei, and so on. Sometimes you actually also see that there are these dividing lines here. So this is a typical example of a so-called a septate fungus, because there are also fungi out there that essentially are so-called aseptate. That means they are long filaments where you cannot see the individual cells, but here you can actually see the individual cells. Just see that, yeah. So these are basically fungi. Those dots that you see here, I suppose that these might be spores. Okay, of the fungus. And then bacteria are the smaller ones over here. Those tiny things that you see here, these are bacteria. They're prokaryotes. So you actually see the size difference quite nicely. That's why one of the reasons why, as a matter of fact, I'm just showing you the same stuff now that I showed my students just on last Friday. This was yesterday actually, because we started off with cell theory just to demonstrate a little bit the difference in cell size. Yeah, so you see that the eukaryotic cells are significantly larger than those bacteria here. Yeah, so that is actually therefore a nice example here. And on those bacteria in the background that you see, very common on cheese Brevibacterium. And what they do is they produce those substances. So they break down the cheese protein, the milk protein, and they release certain substances that give the cheese the typically smelly characteristic. And yes, this is also the next video that I'm going to publish. Why does cheese smell like sweaty feet? I'm going to give you the answer right away, because of course the same bacteria are present and they produce the same waste products, and that's why cheese and sweaty feet smell so similar, because it's actually the same bacteria. Yeah. So that's a little bit something I just wanted to also share with you here. And and you see that the surface of a cheese is a fairly complex, I would say almost ecosystem. Yeah? So what I'm going to do now is because I do slowly have to already finish the live stream. I know it's shorter than before. But I do want to have a look at a couple of questions or comments that you might have. So it's probably, so there are some comments going on here. It's fine. Okay, there's nothing for me. So discussion about pond water. Yeah, as long as it doesn't go anaerobic or moldy, it's relatively safe. Okay, I cannot follow the discussion yet. Yeah. What about having a terrarium set up from pond water? That's fine in terms of biosafety. As a matter of fact, I do have a mini. I have a jar, several jars of pond water here, actually on my side, like a mini aquarium. And honestly, yeah, I don't have a huge problem with that. I will tell you where the problems are. What you have to avoid is you have to avoid so-called fecal bacteria, basically sewage bacteria. You should not have them in your water sample. So usually when they're testing water quality in ponds and also swimming pools and so on, they're usually testing for the presence of so-called fecal bacteria, basically bacteria like E. coli and so on that can be found in sewage. Because if they are present, then that's not so good, right? Otherwise, pond water is of course a complex, lots of microorganisms and so on. But generally, in a pond water, you should not have too many bacteria, because the larger microorganisms, the ciliates, for example, paramecium, and all of these worms and so on, they should actually keep the bacterial load fairly low. And also, if there are too many bacteria present in a water sample, then the oxygen level is too low, right? And then the higher microorganisms like the ciliates cannot survive. So generally, from a bacterial standpoint, pond water or also mini aquarium should not be such a huge problem. Yeah, some people also historically maybe you've heard of what's called a hay infusion, where you take dried grass and you basically put water to it, kind of like and then you allow bacteria to grow. That is a different case because there are lots of bacteria there, and that's actually maybe even possibly a little bit risky. Yeah, because simply the bacterial density then is so high. It's like spoiling food, so to say, right? And so I think there is a difference a little bit between which depends a little bit on the amount of organic material present. So in pond water, generally, if it's relatively clean, the amount of organic material should not be too high. Yeah. Basically a hand microtome. That's actually a good point. I'll try it, okay? I have to get the correct paraffin wax, and I will do a little bit of dehydration. One of the reasons why I did not want to do microtome coats with paraffin is because you have to use xylene, which is a little bit not so healthy. But there is also there are also replacement solutions called HistoClear and so on, which are healthier, so to say, or not dangerous. Yeah. So this is a little bit the thing that let me quickly go through. Yeah. So what I have to do, unfortunately, now is I have to say bye already to this week's live stream. Because unfortunately I have to go somewhere. I hope that next week I'll have again a little bit more time. Yeah. I'm going to basically call it a day for today. Still thank you, even though it was a little bit shorter that you joined me. I wish you all the best. And if you have any recommendations or any wishes for me to put under the microscope, I'm always quite happy for recommendations. And I've seen a few recommendations already today in the live stream. Yeah. Okay. All the best. Happy microbe hunting as always, and hope to see you again. Yeah, and yes, ever smaller. Don't forget to recheck the Fairy Shrimp email. Yes, thank you very much. Okay, I'll check. All the best. Bye bye.